Hi, everyone. My name is Linda Isaacson, and welcome to today's EMSL Learn webinar, where we're going to be discussing aerosol research capabilities at EMSL. We're so glad that you could join us today. We will announce the date for the next webinar pretty soon, and more information will be provided through our social media channels via either email and you'll hear all about our upcoming events and other opportunities. So make sure to keep in touch with EMSL by subscribing to our email and social media channels if you haven't already. The links will be provided in the chat. So for today, EMSL Earth scientist Jay Chen is going to provide details on EMSL's new controlled system to generate combustion aerosols for your research. Narun Naharlata will walk you through how to use EMSL's size and time-resolved aerosol collector for lab and field sampling of particles at different atmospheric conditions. And Gregory van de Grift is going to discuss EMSL's multi-modal mass spectrometry capabilities to characterize atmospheric aerosol composition. There will be time at the end of today's webinars for questions, and we encourage you to post your questions in the Zoom chat. Jay, Narun, and Gregory will answer them during the Q&A portion after their presentations. And so with that, we will begin with Jay. Take it away.
Thank you, Linda, for the introduction. Okay. So as Linda mentioned, today I'm gonna discuss the controlled combustion system at EMSL for user to request and use for their project. So before I start my presentation, I want to first acknowledge that this system is designed and based on my PhD work on the mentor of Dr. Rewan Saleh at the University of Georgia, and I made some improvement and modification. So before I go to the detail of the combustion system, I want to provide some background. So carbonaceous aerosols include black carbon and organic carbon have important impact in our climate systems. They can absorbing and scattering light, causing direct overall warming and cooling effect. Also, they can indirectly affect our climate by acting as seed for warm and cold cloud. Moreover, they can affect our ecosystem by affect the photosynthesis and sorry about that. Photosynthesis and the evapotranspiration of the vegetation. So, however, our understanding of this impact are still limited. So, one reason is a major source of carbonaceous aerosols are combustion, and in real life, combustion conditions such as combustion temperature and air-to-fuel ratio is chaotic. For example, like here, this curve represents the emission property of real-life combustion, which can cover few orders of magnitude in their molecular mass, viscosity, light absorption properties, and also volatility. Therefore, there are a lot of effort has been made in the literature to build some chamber to study the combustion aerosol. One example is biomass combustion chamber. It is very useful to understand the biomass combustion emission property under many real-life combustion conditions. However, they still have some limitations. First, typically combustion happen under smoldering and flaming conditions. Therefore, as you can see here, their properties still quite their aerosol property still has quite wide distribution. Also, this is due to the non-uniform temperature and the limited control in air-to-fuel ratio. Moreover, these biomass combustion chamber typically very large and difficult to maintenance. Another example is a soot generator, and you can tell from its name and mainly focusing on black carbon, and also it is limited of fuel types and typically use gas fuel such as propane. So our motivation is to build a controlled combustion system where we can control the air to fuel ratio and the combustion temperature for different types of fuels. And this is the overall image photo of our combustion system. So it contains a combustion system here, and you can see it as the high temperature combustion chamber made with a high temperature array, and also it is surrounded by 2 high temperature heaters, which can go up to 1200 degrees Celsius. So we use a PID controller to control the temperature inside the combustion chamber. And also we use mass flow controller to control air to fuel ratio. So for the liquid, pure liquid fuel, we use bubbler to deliver into a combustion chamber. And the example of pure liquid fuel is toluene. And for mixed liquid fuel such as gasoline and diesel, we can use this atomizer to deliver into a combustion chamber. In current setup, we can also put the biomass inside the combustion chamber and continuously flow air to make it as a semi-batch combustion chamber. And then the emission gonna be conditioned inside of this emission condition system by adding dilution to reduce the particle concentration. So this is the temperature. This is the temperature profile inside the combustion chamber, and as you can see here, when the inside temperature can reach 950 degrees Celsius in about 40 minutes, and then it maintains constant. And when we turn off the heater, the temperature start to decrease slowly at the rate about 100 degrees Celsius per hour. So now I want to show you some preliminary result. So first, the result is from the toluene combustion at the 950 degrees Celsius. We changed the air to fuel ratio from 0.3 to 0.5. And this is the normalized size distribution of emission from 2 air to fuel ratios. So as you can see here, at the constant temperature, slightly increase the air to fuel ratio, we can have a particle emission size distribution increase significantly, with mode increase from about 20 nanometer to about 300 nanometers. We collect particles on the TM grid and use our tailed SEM stage to take the tailed images. As you can see here, at higher air to fuel ratio, we can have a more black carbon, or also known as soot particles in it, and moreover, particle from the high air to fuel ratio has higher height to width ratio, suggests they might have higher viscosity. Moreover, we collect particle on the Teflon filters, and you can see here for particle collected on the low air to fuel ratio, the color is almost invisible, but at high air to fuel ratio, they are much darker, indicate at higher air to fuel ratio, particle has much higher light absorption properties. So the next example is the diesel combustion at a different temperature with constant air to fuel ratio about 16. So this is a time theories of size distribution, and the color bar represents the normalized size distribution. Red line represents the temperature inside the heater. And as you can see here, at the 1,000 degrees Celsius, we have a particle generated from the combustion system with mode around the 20 to 30 nanometer. And once we stop the heater, the particle size decrease. And at about 950 degrees Celsius, we almost lost every particles. So this indicated that our system can be used for engine emission studies to help develop a better pollution emission control strategies. Last example I want to show you is a biomass combustion. So I put a 10-gram wheat grass collected from another collaborative project at the EMSL called the Twins Ecosystem Project. And I continuously flow 0.5 liters per minute air into the combustion system and gradually increase temperature to 950 degrees Celsius. So as before, this is a time series of the size distribution, and the red line is the heater temperature. As you can see here, with increased temperature, we have a peak lot of emission at about 250 degrees Celsius. This could because there are a lot of volatile species evaporate at this temperature and be oxidized inside the chamber and to form particles. And I collected samples to look under the tailed SEM, and you can see here a lot of particles are very flat. That indicate this particle has very low viscosity and could be in the liquid state. After that, the particle size is well correlated with temperature. That the increased temperature, we can see increase of particle size. So this is what I have today, and our next speaker is Nuran.
So, good afternoon, everyone. I'm Nurun Nahar Lata. I'm a postdoctoral researcher at EMSL. Today, I'm going to talk about EMSL's one of the newly developed aerosol research capability, STAC, which is a size and time resolved aerosol collector. So before we start talking about the STAC, I wanted to give some background why we developed the STAC. Basically, we wanted to learn more about the vertical profile of atmospheric particles because studying the vertical profile of atmospheric particles is important because of understanding the atmospheric processes like cloud formation, precipitate formation in the atmosphere. It also very important because we can monitor the air quality, because it is important to get an idea about the pollutant source and also the transport pattern of the pollutants. The vertical profile also affects the climate modeling, because the vertical distribution of aerosol can affect the incoming solar radiation, which affects the temperature and climate pattern. So several challenges in the aerosol community motivated us to develop this new impactor system. So in atmosphere, there are lots of heterogeneity exist. For example, the vertical gradient of the aerosol composition is exist. Like aerosol composition can be varied from different altitude because of the stratification of the aerosol layer, and different aerosol layer might have different aerosol physicochemical properties. And sometimes the particles at the ground level cannot fully represent the vertical profile. So in the right, I showed our recently published paper cover art where we probe the vertical variability of the aerosol particles, and we saw that due to the vertical stratification of over the Arctic atmosphere, we see a difference in the chemical composition. And there's another challenge, that is the aerosol particles' physical and chemical properties and optical properties are changing rapidly during the transport time, which is really important to monitor. And with the existing aerosol sampler, it is difficult to sample at multiple altitudes with high time resolution and also with the online measurements. So all these challenges actually motivated us to develop this new size and time resolve at automated aerosol sampler for the unmanned aerial system. So to probe the vertical profile of atmospheric interaction and processes and get the size resolve and time resolve chemistry of the particles, we developed a new aerosol system, which is a stack system. So this is the schematic of the stack system. And here, the stack is not only sample the aerosol particles, it also record the environmental condition also, and we can also get the online measurements. So stack is integrated with some environmental sensors like temperature, relative humidity, and altitude information we can get. And we can also monitor the online particle concentration using optical particle size distribution. And the particle light absorption properties can be recorded with microethalometer MA200. We can record particle absorption properties at 5 different wavelengths. So here you can see like 20 impactors are integrated in the system. So that we can get a time resolved aerosol characteristics. So we can accommodate 20 impactors in each of the stack system, and this can be programmed to start at certain time. So there is a computer connected with a impactor system, and we can collect particles at different time resolution. And each of the impactor design is like this, and we can collect particles at different stages. So from stage A collect comparatively larger size particle, and in stage E collect comparatively smaller size particle. So this impactor is a 4-stage cascade impactor. So we can fit four stage in this impactor, so that we can get different so that we can get size resolved chemistry of the particles at different environmental condition. So so we can use the different stages based on the research question we wanted to answer. For example, if we wanted to learn more about the new particle formation in the atmosphere, we can focus on mostly the smaller size stages than the larger. So in that way, we can make a educated guess which size card we wanted to use. So in the right panel, it is showing our recently published paper by Cheng et al., where we discuss more detail about the size and time resolve aerosol collector. If you wanted to know more detail about this work, you can visit this paper. And this impactor system is easily deployable with this tethered balloon system, so that we can collect aerosol particles at different altitude. And this impactor system is very lightweight, and it can be deployed with tethered balloon system, or one can do the ground-based measurement as well. So the stack collector samples can be used for different multimodal analysis. So each of the stage we can collect different particles on different substrates. We can collect particles on carbon B film and also silicon nitride substrates. And different substrates can be analyzed with different multimodal offline chemical analysis techniques. So one of the technique is mostly used is computer control scanning electron microscopy with energy dispersive X-ray spectroscopy. So this gives us elemental composition and morphological information, which is very crucial for understanding the aerosol processes in the atmosphere. So with our the computer control SEM system, we can collect more than 1,000 particles in few hours. So the stack collected samples can be used for understanding the multi-phase chemistry in the atmosphere. So we have a capability called Peltier cooling stage interfaced with an inside the environmental SEM chamber. So this can help us to probe the phase state and hygroscopicity of the particles. So we can probe we can look at the particles, whether if it's solid or liquid or semi-solid, mimicking the atmospheric condition. So this cartoon shows the paper published by Cheng et al., where we probe the particles phase state at different environmental condition. So the stack collected aerosol particles can be analyzed with the ice nucleation stage interface inside the ESEM. So here we can probe the cold cloud formation potential of the single particles. In this system, we can mimic the atmospheric condition, like mimic the cirrus cloud and mixed phase cloud condition to probe the ice nucleation efficiency of the single particles collected on the substrate. So here you can see the schematic, the image of the ice particles growing on the single particle, and you can see the progression of the ice formation. The stack collected particles can be analyzed with the TEM. So the TEM is giving us the chemical composition and morphology, also the crystal structure of the particles. So there are several, the stack collected samples can be analyzed with several other surface sensitive techniques like TOF SIMS, which is time of flight, secondary ion mass spectrometry, X-ray photoelectron spectroscopy, and in situ liquid IR SNOM. So this gives the surface chemistry, surface chemical composition of the particles. So here you can see the map of a particle where we can see the existence of carbon, potassium, and sulfur. Lastly, the stack collected samples can be analyzed with the mass spectrometry platform, for example, the NanoDaisy. The mass spectrometry platform gives us the molecular composition and viscosity estimation. So here you can see the mass spectrum of a sample, and so the mass spectrometry capability will be discussed in detail in the next part of the webinar. So the stack system was deployed at ARM-DOE field campaigns. So here I'm showing some of the field campaign names. Here it was deployed in Alaska, STP site, SAIL in Colorado, and Tracer field campaign. So now I want you to give some hint of the analysis that were done for several field campaigns. So in this slide, I'm showing some highlight of the results from the icebox process particle analysis at Olectic Point, Alaska. So the single particle analysis was done using computer control SEM coupled with energy dispersive X-ray spectroscopy. So from the CCSEM data, chemical composition data, we classify the particles into several classes. So here different color indicates different classes of the particle. So we probe the chemistry of the particles collected at two different conditions. One is cloudy condition and one is during the ice fog time. So we found a difference in the size resolve chemistry. So in the cloudy time, we observe a dominance of the sodium-rich particles. And in the ice fog time, we saw that the particle population are dominated by sulfate and carbonaceous. And interestingly, this observation we found from the ground side. Now I wanted to give some more interesting result, which shows the vertical gradient of aerosol properties at SGP site during the winter time. So this is the this figure shows the flight pattern of the TBS flight, and here green color shows the CPC, like condensation particle counter concentration, which was deployed with the tethered balloon system. So the CPC is an ARM instrument, and the other two color bar shows the temperature and relative humidity. These are obtained from the IMET, which is a ARM instrument as well. And here, if we look at the flight pattern, we can see a gradient of the concentration, temperature, and relative humidity. And we can see in the at lower altitude, we can see comparatively low concentration, and it varies from altitude to altitude. And for relative humidity wise, we can see at high altitude, we can see comparatively high relative humidity. And temperature wise, we can see that temperature is also varying with altitude. And we can see the these are the size resolved chemical composition obtained at different altitude. So the shaded region is showing the all over sampling time. So we can see a variation in the size resolved chemical composition, but though the chemical composition is not significantly different, but we can see distribution. We can see a difference in the distribution. So now I wanted to give a hint of the particle analysis during the tracer campaign, and this work is in collaboration with Alison Steiner from University of Michigan and Alan Goldstein from UC Berkeley. So this shows from this study, we can see a variation in the size resolved chemical composition from altitude to altitude. We can see a difference distribution of the particle size. Particles here, in this case, particles are dominated by carbonaceous in the smaller size range. And we can see the sulfate particles are dominated in the larger size range. Now I wanted to give some hints of the chemical analysis for the SAIL TBS campaign. And this work is in collaboration with Dr. Alex Luskin from Purdue University and Dr. Paul Dimot from University of Colorado. So here we can see we don't see much variation in the size resolved chemical composition, but we can see a difference in the size of the particles, because size can play a very significant role in the cloud particle formation. So with that, I would like to thank all the audiences, and I wanted to give the floor to Gregory Vandergrift, who will talk about the EMSL's mass spec capabilities. Thank you.
Excellent. Thank you, Narun Nahar. Hi, everyone. My name is Gregory Vandergrift, and I'm very excited to talk to you broadly about EMSL's mass spectrometry capabilities for atmospheric research questions. I want to just to give you an overview of what we're going to talk about, just highlight the different kinds of mass spectrometry tools that we have, not necessarily explaining in detail how they work individually, but giving you a broad catalog of the different things at our disposal to help answer your specific research questions. And I recognize a lot of the names in the chat. Thank you to those that are familiar faces for coming by. I appreciate that you might already have a very strong sense about the mass spectrometry capabilities at EMSL, but perhaps since the last time you've looked into EMSL or worked with EMSL, we might have gotten some new toys and some new expertise that I'm excited to talk with you about today. And for the end of the presentation, for the sake of questions, I encourage you to think about and ask how some of these new tools that you might not have heard about us having before, how they can be integrated and to help answer your specific research questions. Again, just some very brief overall context for mass spectrometry and why it's important for atmospheric research in particular. This talk's going to be broken down into two specific categories, both of which are key central capabilities that we offer at EMSL. One being our high resolution mass spectrometry platforms. And the key outcome from these platforms as it pertains to atmospheric research is that we can characterize the constituent organic molecular formulae that comprise an aerosol sample. And why that's important, we'll get into in subsequent slides. Another key category of mass spectrometry capabilities that we have here is single particle mass spectrometry platforms. And in particular, what these can tell us is they can give us in situ and real-time physicochemical properties of individual particles. There's a couple other mass spectrometry resources that I wanted to highlight, even though I will not go into detail today on these specific resources, but since EMSL has a broad capability skill set, we also can do inductively coupled plasma mass spec or ICP-MS, as well as isotope ratio mass spec. Again, we won't focus on them as part of this presentation, but they are resources that we can offer to help answer any research question. What kind of mass spectrometers does EMSL have? We have a vast array of Orbitrap mass analyzers, and we specifically have a Velos Orbitrap instrument that's dedicated to aerosol research. We have a vast array of FTICR, or Fourier transform ion cyclotron resonance mass analyzers, ranging in field strengths from 7 Tesla all the way up to our flagship instrument of 21 Tesla. And we recently also have a time of flight mass analyzer that's specifically dedicated to analyzing volatiles, an Agilent 7250 GC-QTOF system. So what can this high resolution mass spec platforms give us? Specifically, the high resolution and accurate mass from these platforms allows us to have confidence in the assigned molecular formulae. The higher mass resolving power we have, the more we can reduce our ambiguity in the molecular formulae that we assign to these peaks. And once we have these assigned molecular formulae, there's a lot of different things we can do with them to inform atmospheric research questions, such as we can do these parametrizations to inform a volatility distribution. We can inform the viscosity of the constituent compounds, things like phase state, aromaticity, etc. And as an example of this on the right-hand side, this data set was generated in collaboration with Fan May, Dariel Dexheimer, and Maria Zawatowicz. And we have on the left-hand side a volatility distribution of identified molecular formulae in a daytime aerosol sample compared to the constituent organic molecular formulae from a nighttime sample on the right. And what this allowed us to see is in blue, we have a lot more nitrogen containing functional groups in the nighttime compared to the day, which gives us some insight into the kinds of unique chemistries that are happening between these two different periods. So this again is an example of what we can do with parametrizations of our mass spectrometry output. As far as modeling goes, here's an example from where we worked with Ji Zhang and Manish Srivastava, where we took the output from a mass spectrometry data set and we modeled the evaporative kinetics of this particular sample, getting an idea for how this sample might behave over time. Moving forward, I'd like to highlight a few of our mass spectrometry resources specifically, starting with our 21 Tesla FTICR instrument. This field strength is particularly special. It's one of 2 such instruments worldwide. And why you might want to go to such a high resolving power, we alluded to earlier, but the higher the field strength, the higher the mass resolving power, and the higher the confidence you can have in your final molecular formulae assignments. So I wanted to highlight a study here led by Amna Hihaz, who at the time was with Lynn Mazzolini's group. And this figure right here is published in Analytical Chemistry, showing that they were able to use the 21 Tesla FTICR platform to identify organophosphorus compounds in a wildfire-influenced aerosol. These kinds of compounds are particularly difficult to try and identify with lower resolution platforms due to a variety of factors, one of which being abundance, another which of being the lack of another significant isotope of phosphorus. In any case, this is a nice demonstration, and I encourage you to read it, showing how the 21T instrument can be used for specific atmospheric questions. Another example of what this platform can give you, this is a figure from a collaboration with another user, Elizabeth Schenk, where we were able to look at the isotopic fine structure, or looking at the different height of isotopic specific peaks for a formulae that give us increased confidence in the formula that we are assigning to a specific compound. So on the bottom here, you can see some simulated mass spectra with the dashed lines and our experimental mass spectra with the red lines. And our experimental lines up very well with what we see from centroided. All that to say, it's giving us a very high degree of confidence in the final molecular formulae that we are assigning to these peaks. Here's just some other additional photos of some of these mass spectrometry platforms, some of which are certainly newer than others. As highlighted on the left, we have Mickey Rogers working on our GC QTOF, which is one of our newest instruments that we can use to probe the composition of volatiles. On the top right, we have Will Q working on one of our Bruker FTICR mass spec platforms, Narun Nahar with our Aerodyne Aerosol mass spec on the bottom left, and myself and our team lead, Swaroop China, on the bottom right, using our NanoDesi platform on a Thermo Fisher Orbitrap mass spec. Chromatography separations. Again, very standard for a lot of the people in this chat, I'm sure, but I just wanted to highlight the different options, again, that we have available here at EMSL. We have a variety of different LC platforms and a variety of different kinds of ionization sources that we can put on the back end of these separations, including ESI, APCI, APPI, nanospray, and we'll go into those in a little bit more detail later. And we can also use a linear photodiode array detector to try to quantify and detect things that have a chromophore, for example, brown carbon. We also, again, as we see Mickey working on it in this slide right here, a gas chromatography system as part of our new instrument. And this is coupled ideally to this TOF mass analyzer. And why we're particularly excited about this capability, the TOF mass analyzer is particularly fast with respect to acquisition speed. So coupling that with a chromatographic separation is particularly ideal because the fast acquisition speed will let us capture even some of these smaller, exceedingly transient chromatographic peaks. So if you have research questions that do with volatile composition, I think we have a great brand new capability to help address that. Moving into our types of ionization sources, and I've sorted them into two categories here, one being ambient ionization sources and vacuum ionization sources. So first, looking at the varieties of ambient ionization sources we have, again, for a lot of us in this seminar, this won't be terribly news-breaking or new information, but again, I wanted to highlight the different things that we have available to users at EMSL. We can use electrospray ionization, ESI, we can use atmospheric pressure chemical ionization or photoionization, which are APCI and APPI respectively. And we have on the bottom left of the slide just a general diagram that helps inform which ionization source might be useful in different scenarios, specifically across different molecular masses and different polarities of analytes. And an example of why this might be helpful for atmospheric research, here's a paper from Lynn Mazzolini's group that worked with us in the past, showing that for a particular aerosol sample, they generated a composite data set using APPI and electrospray ionization across different ionization modes. And the different colors here correspond to different unique molecular formulae that were detected with different ionization sources. So again, depending on your research question, we might use a specific source as the first line, but because we have all these different availabilities, we can generate a more comprehensive data set at the end of the road. We also have some different ionization sources that are typically under vacuum. I apologize, the acronym's not correct on the first one for laser desorption ionization, that should say LDI, but both LDI and MALDI are other capabilities we can use for aerosol-related samples, and they're typically a lot better for things that are, again, less polar, non-volatile compounds that can absorb the incident radiation from the laser ablation process. We also have electron ionization, which is the ionization source that we use on our GC QTOF instrument. And on the right-hand side, again, from working with Lynn Mazzolini and Amna Ihaz, we have an example of what laser desorption ionization looks like for a biomass burning sample. So I have included on the top left-hand there a picture of what the aerosol substrate looks like before and after. So you can see that we have less sample loading after because the incident laser has helped to volatilize a lot of our samples so that we can see it on the right-hand side. And again, plotting double bond equivalence versus number of carbons, highlighting the degree of complexity that we can see for these different samples. The one resource that I wanted to spend perhaps a little selfishly a little bit more time on is nanospray desorption electrospray ionization. This is the capability that I spend the most time with here at EMSL. And what this allows us to do is we can directly analyze an aerosol filter from a substrate that it's collected on. The picture on the left-hand side, you can see this is interfaced right in front of the inlet of a mass spectrometer. It consists of these two silica capillaries that are aligned at approximately 90 degrees, and a solvent junction is then used to desorb and then sequentially ionize aerosol sample right from the filter itself. The picture on the right-hand side gives you perhaps a better idea about the kinds of aerosol samples that we can apply this to. That dashed red line down the middle is actually a deposited aerosol sample. You might have to squint your eyes to see it, but thankfully the NanoDesi platform is sensitive enough to desorb even sample loadings as minimal as this. Essentially, this very small, very concentrated in situ extraction helps us have this very high sensitivity. Certainly, direct analysis might have its caveats, but NanoDesi is very highly advantageous with respect to ease of sampling and consequent sensitivity for the samples that it can be applied to. And our previous presenter, Narun Nahar-Latta, highlighted our stack platform, which is a really unique capability for collecting samples. We can also collect bulk aerosol as part of the stack platform. And that picture on the top right-hand side is actually the use of NanoDesi to analyze aerosol collected from the stack platform. So again, very dilute, very low loadings, but because the NanoDesi system allows for this concentrated extract, we can still see useful results. NanoDesi you can think of very similar to electrospray ionization with respect to molecular coverage. And as a result, we can kind of tailor the sampling solvent systems we use to try to push the molecular coverage in a particularly direction of interest. And especially notable, this NanoDesi platform can be interfaced with any of our mass spec systems, including Orbitrap and FTICR. So I wanted to just spend a little bit of time on this slide, just showing some examples of the data that we've gotten from interfacing nanospray desorption electrospray ionization with our high resolution mass spec. On the left is from a collaboration with Fan Mei and Dariel Dexheimer. This is showing the analyzing the aerosol molecular composition of an aloft sample collected with the stack platform versus one collected at ground level. The things that are more transparent were common with the ground level, whereas the things that are colored, we uniquely detected in aloft. So again, facilitated by the sensitivity of NanoDesi, we can see some interesting, unique features. This middle plot here, again, with Fan Mei, Dariel Dexheimer, and Maria Sawa-Wadaj, we looked at how wind direction can affect the molecular composition. So on the left, we have fairly uniform winds, where on the right-hand side, that sample was very southerly focused, collected from southern Great Plains in Oklahoma. And as a result, you can see these different volatility distributions from these different samples facilitated by the NanoDesi high-resolution mass spec platform. And finally, on the right-hand side, something that we've worked on with Lynn Mazzolini, anything that we acquire with our mass spec platforms can be informed in a very complementary way by our microscopy platforms. So I won't get into the details specifically of this figure, but I wanted to present it to show that STIXM NextSath, something touched on by Narun Nahar, as well as computer-controlled scanning electron microscopy, all capabilities that we offer at EMSL can be used in a complementary fashion to our other mass spec-based capabilities. And I'll briefly spend some time on this. I'm not an expert on it. Thankfully, we have one here at EMSL, Ala Zelenyak, our single particle mass spectrometry platform, or mini-splat. It's pictured on the top left-hand side there, and we have Ala on the bottom left. What this allows us to do is get in situ in real-time physicochemical properties of individual particles. And it gives us a host of different kinds of information. And as you can see on the bottom center, there's a representative mass spectrum for a biomass burning aerosol, and some other sample-specific types on the bottom right. It can also inform volatility of different size-selected secondary organic aerosol, and this is a very robust and field-deployable instrument. So just to quickly finish off here, I just wanted to have a small slide about why EMSL is a great place to collaborate with. Hopefully these last presentations have given you an idea as to why that is, but specifically we have all these capabilities in one place. So when it comes to shipping samples all over the place, a lot of the times we can do a lot of these complementary analyses across the hall as opposed to in a completely different facility. I want to highlight the team headed by Sarup China on this slide. You've met Jay Chang and Naruna Harlata through our previous talks, but we have Mickey Rogers and Tanya Gautam, who are some new additions who focus on bioaerosol and secondary organic aerosol, respectively. Users can come on site where we have a highly collaborative atmosphere. And because there's a lot of different capabilities here at EMSL, we have a lot of opportunities to learn, expand, and grow into different additional research spaces besides just atmospheric chemistry. And in closing, we have a number of different proposal calls that we have detailed here on the slide. The QR code on the bottom left will take you to our proposal webpage. But if you'd like to have any clarification, please email any of us. Our emails are here on the slide. We'd be happy to help guide your research questions to the best place. And we look forward to hopefully and potentially working with many of you in the future. Thank you very much. And with that, I will hand it over to Linda.
Wonderful. Thank you, guys. So we are moving into the question and answer section for this webinar. So I guess, yeah, if Gregory, Narun, Jay, can you go ahead and turn your cameras back on? We will get started. And Heather, if you can go ahead and spotlight them, that would be great. So first question that we've received from the audience. So this question is directed to Jay. It's from, I believe his name is pronounced Saikat Goosh, but don't be upset with me if I misspelled or mispronounced your name. He asked, or he said, "Hi Jay, great talk. Can you perform chemical analysis of the particles from the combustion chamber, like organic carbon, elemental carbon, sulfates, nitrates, etc.?"
Thank you for the question. The short answer is yes. So as Greg mentioned, we can connect the either online aerosol mass spectrometers or collect filter for offline mass spectral analysis. Since our combustion system is like a combustion aerosol generator, so it can be used for both online and offline analysis.
Great. All right. So next question. Actually, this is going to be a non-science question. So, and Gregory, you might be able to answer this question best, since you went over the different proposal opportunities that are available at EMSL. We did have a question about how to provide more information on or if you could provide more information on the request process for EMSL for access to these instruments.
Yeah, thanks. That's a great question. And I will, after I finish saying my piece, defer to Jay and Narun Nahar to add any additional details as well. The first thing that I would suggest is to simply get in contact with any of us at EMSL. For the best crafted proposals, it's one that we spend a little bit of time on to make sure the research question is focused and well-defined, and one, to make sure that it's in line with some of the focus areas that EMSL is hoping to fund. So for that reason, I encourage and direct people to go to the EMSL website to see the focus areas, as well as for the upcoming funding cycles that might align with their timeline and research interests. But beyond that, again, I'd encourage people to send emails to people like myself or Jay or Narun Nahar, because it might be that you have a really great research question, but you are unsure of the extent or the full extent of resources or the extent of the capabilities of those resources that we have at EMSL. And starting those conversations with us can help guide how those resources can be best used to answer the questions. And once that is better defined, those are the proposals that are typically the strongest and the most well-received. So specifically, it usually starts with things like letters of intent, and those are, in my opinion, best crafted, not alone, but rather with us. You don't have to have something accepted to start this letter of intent progress. It's something that's best facilitated, a bit of a back and forth conversation between us and you to make sure that we have a great direction together, because it's meant to be collaborative. So why not start that collaboration from day one? Jay, maybe first to you and then to Narun Nahar after that, if you have something to add.
Yeah, I think Greg provided very comprehensive information. So I just want to add a few points. First, every year we will have different core topics, and that gonna based on the like any emerging research scientific question, and usually the large scale proposal call and EMSL FICUS proposal call gonna be open, announced and open at later December every year. And like Greg mentioned in his presentation, the LOI gonna typically do it at during the March time. And if once it proposed, and I also encourage if any of you have any question or have a good scientific ideas, but not sure how we can help, you can always come to reach to us. And also our team leader, China. Yes, don't hesitate to bother him. Yeah.
Narun, did you have any? Okay.
Yeah, I have issue with unmuting myself. Yeah, Gray and Jay both gave us I think the most of the comprehensive idea about for the proposal call. So yeah, so I just wanted to say in the same voice, like if you have any idea and don't know which instruments you want need to write, just reach us. We can help you, like based on the research question you wanted to pose. Yeah, so don't hesitate to contact us if you have any need anything from us. Yeah.
All right, so back to the science questions. Eric Hill is asking, will you talk more about the optical-based instrument you use to measure and to quantify aerosol particle sizes? So I don't know, is that yours, Narun?
Maybe I can take this question. So to match, I assuming you are asking about the like particle size measurement capability at the EMSL. So we for the stack we can add an optical particle counter to measure the size distribution, and also we on the ARM tether bloom system it deployed other particle size measurement instrument like POPs. Sorry, I forgot the full name. And yeah, no, you can add that.
Yeah, it's portable optical particles spectrometer.
Yes, and for the size particle size measurement at EMSL, we have a SMPS. Hold on. Scanning mobility particle size, yes, and as well as OPC, optical particle sensor, yes, and these are the instrument we have at the EMSL, and also I think as Greg mentioned, in Ala's lab we have another lot of particle size classifiers, and these can also be requested when you submit a proposal.
Linda, we cannot hear that particular question.
Sorry, I had muted myself too. And anyway, so Narun, can you tell us, can you do laboratory studies using STAC?
Yes, we can do laboratory study like with a standard material, also like laboratory generated aerosol, we can do that. Yeah, we have we have a different pump which we can connect to collect particles in the atmosphere, like in the laboratory setup as well. Yeah.
Okay, and a follow-up question for you as well. So Yu Yao asked, great talks, or said, great talks, y'all. I have a question for Narun. Are there any differences in chemical composition